Welcome to the Life Science Coach, where biology comes alive. Have you ever wondered how millions of tiny cells inside your body work together to keep you alive every second? In this episode, we dive deep into the fascinating world of cell biology, the science of life at its most fundamental level. From the powerhouse mitochondria to the intelligent nucleus controlling every function. You'll discover how cells grow, communicate, divide, and sustain life. We'll begin with a simple introduction to cells and understand why they are called the building blocks of life. Then, we'll travel through the history of cell discovery, from early microscopes to modern biology. Next, we'll understand the famous cell theory that explains how all living things are made of cells. Finally, we'll dive into important cell organelles and discover how each part of the cell performs specialized functions. Before modern biology evolved, scientists began uncovering the hidden world of cells through simple microscopes. In the 17th century, Robert Hooke first used the term cell while observing cork under a microscope. Later, Anton van Leeuwenhoek discovered tiny living organisms, which he called animalcules, opening the door to microbiology. By the 1830s, scientists realized that cells were the fundamental units of life. Matthias Schleiden concluded that all plants are made of cells, while Theodor Schwann proved the same for animals, forming the foundation of cell theory. Finally, Rudolf Virchow stated that new cells arise only from pre-existing living cells, a principle that transformed modern biology forever. Cell biology is one of the most important branches of biological science because it helps us understand life at its most basic level, the cell. It focuses on the study of cell structure, function, molecular organization, growth, reproduction, and genetics. In simple terms, cell biology explains how cells work, communicate, divide, and perform specialized functions inside living organisms. Every tissue, organ, and system in our body depends on the proper functioning of cells. Every living organism, from tiny bacteria to complex humans, is made up of cells. The word cell comes from a Latin term meaning small room, first used to describe tiny compartments observed under a microscope. Scientists later defined the cell as a living unit surrounded by a semipermeable membrane and capable of self-reproduction. Cells are highly organized and contain genetic information that controls their activities. They can produce energy, perform chemical reactions, respond to stimuli, and even reproduce themselves. In short, cells are the fundamental building blocks. Cells are mainly classified into two major types, prokaryotic cells and eukaryotic cells. Prokaryotic cells are simple and primitive cells that do not contain a true nucleus. Bacteria are the most common examples of prokaryotic cells. On the other hand, eukaryotic cells are more advanced and contain a well-defined nucleus along with membrane-bound organelles. Eukaryotic cells are further divided into plant cells and animal cells. Plant cells contain structures like the cell wall and chloroplasts, while animal cells are specialized for movement and complex body functions. Cells are broadly divided into two major categories, prokaryotic cells and eukaryotic cells. Let us understand the important differences between them. Prokaryotic cells are smaller and simpler in structure, usually ranging from 1 to 10 micrometers in size. They are generally unicellular organisms, meaning the entire organism consists of just one cell. In contrast, eukaryotic cells are larger and more complex, ranging from 10 to 100 micrometers, and are commonly found in multicellular organisms like plants and animals. One of the most important differences is the presence of a nucleus. Prokaryotic cells do not have a true nucleus, whereas eukaryotic cells possess a well-defined nucleus enclosed by a nuclear membrane. The genetic material in prokaryotes is circular DNA and lacks histone proteins. But in eukaryotes, DNA is linear and associated with histones for proper organization. Prokaryotic cells also lack membrane-bound organelles such as mitochondria, endoplasmic reticulum, and Golgi bodies. Eukaryotic cells, however, contain all these specialized organelles that perform different cellular functions. Another major difference lies in ribosomes. Prokaryotes have smaller 70S ribosomes freely floating in the cytoplasm, while eukaryotes contain larger 80S ribosomes often attached to the endoplasmic reticulum. When it comes to cell division, prokaryotic cells divide by a simple process called binary fission, whereas eukaryotic cells divide through mitosis. Examples of prokaryotes include bacteria and rickettsia, while fungi, plants, and animals are examples of eukaryotic organisms. The structure of a prokaryotic cell, one of the simplest and oldest forms of life on Earth. Prokaryotic cells are unicellular organisms that lack a true nucleus and membrane-bound organelles. They are commonly found in bacteria and cyanobacteria. Even though they are simple in structure, prokaryotic cells perform all essential life processes efficiently. Let's understand the important parts of a prokaryotic cell. The outermost layer is the capsule, which protects the cell from harsh environmental conditions and prevents dehydration. Beneath the capsule lies the cell wall, providing shape, support, and protection to the cell. Inside the cell wall is the plasma membrane, which controls the movement of substances in and out of the cell. The cell contains tiny structures called ribosomes, which are responsible for protein synthesis. Unlike eukaryotic cells, the genetic material is not enclosed inside a nucleus. Instead, the DNA is present freely in a region called the nuclid. Some prokaryotic cells also possess a flagellum, a whip-like structure that helps in movement and locomotion. Eukaryotic cells, the more advanced and complex type of cells found in plants, animals, fungi, and protists. Unlike prokaryotic cells, eukaryotic cells contain a true nucleus enclosed by a nuclear membrane and several membrane-bound organelles that perform specialized functions. Let's first look at the animal cell. Animal cells are generally irregular or round in shape and do not possess a cell wall. They contain important organelles such as the nucleus, mitochondria, Golgi complex, lysosomes, ribosomes, endoplasmic reticulum, and centrioles. The nucleus acts as the control center of the cell and contains genetic material. Mitochondria are known as the powerhouse of the cell because they produce energy in the form of ATP. The Golgi complex modifies and packages proteins, while lysosomes help in intracellular digestion and waste removal. Now let's examine the plant cell. Plant cells are usually rectangular in shape due to the presence of a rigid cell wall, which provides strength and protection. One unique feature of plant cells is the presence of chloroplasts, which contain chlorophyll and carry out photosynthesis, the process by which plants prepare their food using sunlight. Plant cells also possess a large central vacuole that stores water and helps maintain cell pressure. Both animal and plant cells contain membrane-bound organelles and perform essential life functions, but their structures differ according to their roles in living organisms. Although both animal and plant cells are eukaryotic cells, they differ in structure and function based on the needs of the organism. Let's compare them one by one. Both animal and plant cells contain important organelles such as the nucleus, mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, cytoplasm, and cell membrane. The nucleus acts as the control center of the cell and stores genetic material. Mitochondria produce energy required for cellular activities, while ribosomes help in protein synthesis. Now let's look at the major differences. Plant cells possess a rigid cell wall outside the cell membrane, which provides shape, support, and protection. Animal cells do not have a cell wall, making them more flexible in shape. Plant cells also contain chloroplasts, which carry out photosynthesis and help plants prepare food using sunlight. Animal cells lack chloroplasts because animals obtain food from external sources. Another important difference is the vacuole. Plant cells usually have one large central vacuole for storing water and maintaining turgor pressure, whereas animal cells contain smaller vacuoles. Animal cells contain centrioles, which help in cell division. These structures are generally absent in higher plant cells. In terms of shape, animal cells are usually irregular or round, while plant cells appear more rectangular due to the presence of the cell wall. A cell is made up of two major parts, the hyaloplasm, also known as cytoplasm, and several specialized structures called cell organelles. These organelles work together like tiny machines to keep the cell alive and functioning properly. Some important cell organelles include the cell wall, plasma membrane, endoplasmic reticulum, ribosomes, Golgi bodies, lysosomes, chloroplasts, mitochondria, centrioles, and the nucleus. Each organelle performs a specific function inside the cell. The cell membrane controls the movement of substances in and out of the cell. The endoplasmic reticulum helps in protein and lipid synthesis, while ribosomes are the sites of protein production. The Golgi bodies modify, package, and transport proteins to different parts of the cell. Lysosomes act as the digestive units of the cell by breaking down waste materials. Mitochondria are known as the powerhouse of the cell because they generate energy in the form of ATP. In plant cells, chloroplasts carry out photosynthesis and help prepare food using sunlight. The nucleus serves as the control center and stores the genetic information of the cell. These tiny structures are called organelles, meaning little organs, because each one performs a specialized function necessary for survival. Membrane-bound organelles are mainly found in eukaryotic cells, making them more organized and complex than prokaryotic cells. Everything inside a cell except the nucleus is collectively called the cytoplasm. It is a clear, jelly-like, watery substance that surrounds and supports all the cell organelles. The cytoplasm acts as the working environment of the cell where many important life processes occur. In terms of composition, cytoplasm contains about 90% water and around 10% organic and inorganic compounds, including proteins, salts, sugars, and enzymes. Let's look at its major functions. First, the cytoplasm helps maintain the shape and consistency of the cell. Second, it serves as the medium where important chemical reactions required for metabolism take place. Many metabolic pathways occur inside the cytoplasm, including glycolysis, which is the first step of cellular respiration. It also plays an important role in protein synthesis, fatty acid synthesis, and purine synthesis. Plant cells are surrounded by a strong, non-living, and rigid outer covering called the cell wall. The cell wall provides shape, strength, support, and protection to the cell. It also prevents the cell from bursting due to excess water intake. Unlike the plasma membrane, the cell wall is freely permeable, allowing substances to pass through easily. The structure of a differentiated plant cell wall consists of three major layers. The first layer is the middle lamella. It acts like a cementing layer that joins adjacent plant cells together. The middle lamella is mainly composed of calcium and magnesium pectate. The second layer is the primary cell wall. It lies between the middle lamella and the plasma membrane. This layer is flexible and growing cells usually possess a well-developed primary wall. It is mainly composed of hemicellulose, cellulose, pectin, proteins, and lipids. The innermost layer is the secondary cell wall. This layer is thicker, stronger, and mainly composed of cellulose. It may also contain substances such as lignin, suberin, and cutin, which provide additional rigidity and waterproofing. The cell wall is absent in animal cells but is present in plants, fungi, and many bacteria. The cell wall is a strong, rigid outer covering that provides shape and structural support to the cell. One of its major functions is protection. It protects the cell from mechanical injury, harmful microorganisms, and environmental stress. The cell wall also prevents the cell from bursting when excess water enters through osmosis. Another important function is maintaining the shape and rigidity of plant cells, helping plants remain upright and stable. The cell wall is freely permeable, allowing water, minerals, and gases to pass through easily. It also helps in cell-to-cell communication through tiny channels called plasmodesmata. In many plants, the cell wall stores substances such as carbohydrates and provides additional strength through compounds like lignin. The cell wall also plays a major role during plant growth and development by controlling cell expansion. The plasma membrane forms the outer boundary of the cell and separates the internal environment from the external surroundings. It is mainly made up of a phospholipid bilayer with protein molecules scattered throughout it, forming what scientists call the fluid mosaic model. In terms of composition, the plasma membrane contains approximately 60% lipids and 40% proteins. Let's understand its structure. The phospholipid bilayer consists of two parts, hydrophilic heads that attract water and hydrophobic tails that repel water. This arrangement helps maintain the membrane stability and selective permeability. Embedded within the membrane are different proteins such as integral proteins and peripheral proteins, which help in transport, communication, and recognition. Some membranes also contain glycoproteins, glycolipids, and cholesterol, which provide flexibility and stability. Now let's discuss the major functions of the plasma membrane. Its most important function is regulating the movement of substances into and out of the cell. It selectively allows essential molecules like nutrients, water, and gases to enter while removing waste products from the cell. The plasma membrane also acts as a protective barrier between the intracellular and extracellular environments. In addition, it helps in cell signaling, communication, and maintaining the internal balance of the cell, known as homeostasis. Mitochondria were first observed by Kolliker in 1850 in muscle cells, where he called them sarcosomes. Later, Altman studied these structures in detail and named them bioblasts. Finally, in 1898, Bender stained these organelles with crystal violet and gave them the name mitochondria. Mitochondria are found in almost all eukaryotic cells and are mainly responsible for producing energy in the form of ATP through cellular respiration. Let's understand their structure. Mitochondria possess a double membrane structure. The outer membrane surrounds the entire organelle and contains special proteins called porins that allow substances to move in and out. Between the two membranes lies the intermembrane space, which contains ions and molecules similar to the cytosol. The inner membrane is highly folded and forms structures called cristae. These folds increase the surface area for ATP production and contain enzymes required for cellular respiration. Inside the inner membrane lies the matrix, which contains enzymes, mitochondrial DNA, ribosomes, and other molecules needed for energy metabolism. An interesting fact is that mitochondria have their own DNA and ribosomes, supporting the endosymbiotic theory. Because they generate most of the cell's energy, mitochondria are especially abundant in highly active cells such as muscle cells. Mitochondria are mainly composed of proteins, which make up about 70% of their structure. They also contain around 25 to 30% lipids, while small amounts of RNA and DNA are also present. An interesting feature of mitochondria is that they possess their own DNA and ribosomes, allowing them to synthesize some of their own proteins. Now let's discuss the functions of mitochondria. The most important function of mitochondria is the production of ATP, which is the energy currency of the cell. Because of this, mitochondria are popularly known as the powerhouse of the cell. The Krebs cycle, an important stage of cellular respiration, takes place inside the mitochondrial matrix. Mitochondria also play a vital role in regulating cellular metabolism and maintaining energy balance within the cell. They are involved in several important metabolic pathways such as the TCA cycle, fatty acid oxidation, and parts of the urea cycle. Mitochondria are oval or rod-shaped organelles surrounded by a double membrane. The outermost layer is called the outer membrane. It is smooth and contains special proteins that allow molecules to move in and out of the mitochondria. Inside the outer membrane is the intermembrane space, which separates the outer and inner membranes. The inner membrane is highly folded inward to form structures known as cristae. These folds increase the surface area, allowing more space for enzymes involved in ATP production. Embedded on the cristae are ATP synthase particles, which help synthesize ATP during cellular respiration. The innermost region of the mitochondria is called the matrix. The matrix contains enzymes, ribosomes, granules, mitochondrial DNA, and molecules necessary for metabolic reactions such as the Krebs cycle. One fascinating feature is that mitochondria possess their own DNA and ribosomes, which means they can produce some of their own proteins independently. Because mitochondria generate energy required for almost every cellular activity, they are known as the powerhouse of the cell. Chloroplasts are present only in plant cells and certain plant-like protists. They contain a green pigment called chlorophyll, which gives plants their characteristic green color. More importantly, chloroplasts perform photosynthesis, the process by which plants convert sunlight into chemical energy and prepare food. Because of photosynthesis, chloroplasts are considered the ultimate source of organic compounds and energy for life on Earth. An average plant cell may contain around 20 to 40 chloroplasts, depending on its function and activity. In organisms like Chlamydomonas, a single chloroplast may be present in each cell. Now let's look at the chemical composition of chloroplasts. Chloroplasts contain approximately 50 to 59% proteins and about 21 to 34% lipids. They also contain chlorophyll pigments, which make up around 5 to 8% of the composition and are essential for capturing sunlight. Other pigments called carotenoids are also present and help in light absorption and protection against photodamage. In addition, chloroplasts contain RNA and DNA, showing that they possess their own genetic material similar to mitochondria. Let's understand the origin, evolution, structure, and functions of chloroplasts. Scientists believe chloroplasts evolved from ancient photosynthetic bacteria called cyanobacteria through a process known as endosymbiosis. According to the endosymbiotic theory, an early eukaryotic cell engulfed a cyanobacterium, and over time it became a permanent organelle called the chloroplast. Chloroplasts later diversified into different types of plastids specialized for various functions. For example, chromoplasts are responsible for color pigments in fruits and flowers. Amyloplasts help in starch storage. Etioplasts are involved in light-dependent development, while proplastids are immature plastids that develop into other plastid forms. Now let's look at the structure of chloroplasts. Chloroplasts are usually plano-convex or lens-shaped organelles measuring approximately 5 micrometers by 2 to 3 micrometers. They possess a double-layered membrane envelope consisting of an outer and inner membrane. Inside the chloroplast is a fluid-filled region called the stroma or matrix. Embedded within the stroma is the lamellar system, made up of flattened membranous sacs called thylakoids. Stacks of thylakoids form structures called grana. These grana contain chlorophyll pigments and are the main sites of the light reactions of photosynthesis. Chloroplasts also contain plastoglobuli, ribosomes, enzymes, RNA, and their own DNA. Now let's discuss the functions of chloroplasts. The primary function of chloroplasts is photosynthesis, where sunlight, carbon dioxide, and water are converted into glucose and oxygen. Chloroplasts also contribute to cytoplasmic inheritance because they contain their own genetic material. The process of energy transduction in chloroplasts, also known as the light-dependent reactions of photosynthesis. This process takes place in the thylakoid membrane of the chloroplast and converts light energy into chemical energy in the form of ATP and NADPH. The process begins in photosystem II, where photons of sunlight strike the chlorophyll molecule called P680. This excites electrons to a higher energy level. To replace these lost electrons, water molecules undergo photolysis, splitting into oxygen, hydrogen ions, and electrons. As a result, oxygen is released as a byproduct. The excited electrons move through the electron transport chain via plastoquinone and the cytochrome B6F complex. During this movement, hydrogen ions are pumped into the thylakoid lumen, creating a proton gradient. Next, the electrons reach photosystem I, where another photon excites them again at the P700 reaction center. These high-energy electrons are finally transferred to NADP+, forming NADPH with the help of the enzyme ferredoxin NADP reductase. Meanwhile, the proton gradient generated across the thylakoid membrane drives the ATP synthase complex. As hydrogen ions flow back through ATP synthase, energy is released to convert ADP into ATP. Thus, the light-dependent reactions produce two important energy molecules, ATP and NADPH, which are later used in the Calvin cycle to synthesize glucose. The thylakoid membrane contains several important protein complexes responsible for the light reactions of photosynthesis. These include photosystem II, cytochrome B6F complex, photosystem I, and ATP synthase. The membrane is organized into two regions, the oppressed region, where thylakoids are stacked to form grana, and the non-oppressed region, also called the stroma lamellae. Photosystem II is mainly concentrated in the oppressed regions of the grana. It captures light energy and initiates the process of electron transport by splitting water molecules. This process releases oxygen, hydrogen ions, and electrons. The electrons then move to the cytochrome B6F complex, which acts as an electron carrier and helps pump protons into the thylakoid lumen. This creates a proton gradient across the membrane. Photosystem I is mostly located in the non-oppressed regions. Here, electrons are re-energized by light and used to reduce NADP+ into NADPH. Another important complex is ATP synthase, usually found in the stroma-facing regions of the membrane. It uses the proton gradient to synthesize ATP from ADP and inorganic phosphate. Together, these complexes work in a coordinated manner to convert light energy into chemical energy in the form of ATP and NADPH. In higher eukaryotic organisms, the nuclear genome is usually diploid and inheritance occurs through sexual reproduction. During meiosis, gametes such as sperm and egg cells become haploid, carrying half the genetic material. After fertilization, the zygote inherits one nuclear allele of each gene from both parents. However, things are different when it comes to certain cell organelles like chloroplasts and mitochondria. These organelles possess their own DNA, known as cytoplasmic genomes. In most plants, chloroplast genomes are inherited mainly from the female parent, a phenomenon called maternal inheritance or cytoplasmic inheritance. This happens because the male gamete often contributes very little cytoplasm during fertilization. In many plants such as tobacco, the chloroplast and mitochondrial genomes present in the male gamete are degraded before fertilization occurs. As a result, the offspring receive chloroplast DNA almost entirely from the mother. This type of inheritance is different from Mendelian inheritance because it does not follow the typical dominant and recessive gene patterns controlled by nuclear DNA. In this diagram, the female parent carries dominant alleles represented as A, B, and C, while the male parent carries recessive alleles represented as A, B, and C. Both parents possess nuclear DNA as well as cytoplasmic organelles like chloroplasts and mitochondria. During gamete formation, the female gamete retains a large amount of cytoplasm containing chloroplast and mitochondrial genomes. However, in many organisms, especially plants, the male gamete contributes mainly nuclear DNA and very little cytoplasm. As fertilization occurs, the zygote receives one allele from each parent, resulting in combinations such as AA, BB, and CC. This demonstrates normal Mendelian inheritance for nuclear genes. But when we look at the cytoplasmic genomes, represented here by chloroplasts and mitochondria, they are inherited almost entirely from the female parent. The organelles present in the male gamete are usually absent or degraded before fertilization. This is why chloroplast and mitochondrial inheritance is often called maternal inheritance or cytoplasmic inheritance. The diploid zygote therefore contains nuclear genes from both parents but cytoplasmic genomes primarily from the mother. This concept is important in plant genetics, evolutionary biology, and studies involving organelle DNA inheritance. A classic example of cytoplasmic inheritance using the 4 o'clock plant, scientifically known as Mirabilis jalapa. This experiment demonstrates how chloroplast DNA influences leaf color and how chloroplasts are inherited mainly from the female parent. In this plant, leaves may appear green, white, or variegated. Green leaves contain normal chloroplasts capable of photosynthesis, while white leaves contain defective chloroplasts lacking chlorophyll. Variegated leaves possess a mixture of both normal and defective chloroplasts. Let's look at the inheritance pattern. When a white female plant is crossed with any male plant, the offspring are usually white because the egg cell contributes defective chloroplasts. Similarly, when a green female plant is crossed with any male plant, the offspring become green due to the inheritance of healthy chloroplasts from the mother. The most interesting case occurs with a variegated female plant. Since its egg cells may contain different combinations of normal and defective chloroplasts, 3 types of offspring can be produced. Some offspring become white, some become green, and some become variegated. This variation occurs because chloroplasts are distributed randomly during cell division. The pollen or male gamete contributes almost no chloroplasts, so leaf color depends mainly on the chloroplasts inherited from the female parent. This experiment provides strong evidence for maternal inheritance and demonstrates that chloroplast genes can influence visible traits such as leaf color. The endoplasmic reticulum is a complex, web-like network of membranes present throughout the cytoplasm of eukaryotic cells. It appears in the form of flattened sheets, sacs, and tube-like structures, creating several membrane-enclosed spaces inside the cell. The term endoplasmic reticulum can be understood by breaking the words. Endos means inside. Plasmic refers to cytoplasm. And reticulums means network. So, the endoplasmic reticulum literally means a network of membranes inside the cytoplasm. The ER is interconnected and consists of structures called cisternae, tubules, and vesicles. One important feature is that the ER is continuous with the outer membrane of the nucleus and also connects functionally with the plasma membrane. This allows efficient transport and communication within the cell. The endoplasmic reticulum plays several important roles such as protein synthesis, lipid synthesis, intracellular transport, and membrane formation. There are two major types of ER. Rough endoplasmic reticulum, which contains ribosomes on its surface and helps in protein synthesis. And smooth endoplasmic reticulum, which lacks ribosomes and is mainly involved in lipid synthesis and detoxification. The rough endoplasmic reticulum, or RER, appears rough because ribosomes are attached to its surface. These ribosomes actively synthesize proteins, especially proteins that are secreted outside the cell or used in membranes. Because of this, the rough ER acts as the major site of protein synthesis inside the cell. The proteins produced here are folded, modified, and transported to other organelles such as the Golgi apparatus. Now let's look at the smooth endoplasmic reticulum, or SER. Unlike rough ER, smooth ER does not contain ribosomes on its surface, giving it a smooth appearance. The smooth ER is mainly involved in the synthesis of lipids, phospholipids, cholesterol, and steroid hormones. In muscle cells, a specialized smooth ER stores calcium ions, which are essential for muscle contraction. Another very important function of the smooth ER is detoxification. Cells in the liver and kidneys use smooth ER enzymes to detoxify drugs, alcohol, toxins, and carcinogenic substances. Smooth ER also participates in glycogen metabolism and helps release free glucose into the bloodstream. In addition, the endoplasmic reticulum serves as an internal transport system of the cell, helping circulate materials between different organelles. It also provides a large surface area for various enzymatic reactions and helps in storage of proteins and minerals. The Golgi complex consists of a series of flattened, membrane-bound sacs called cisternae. Usually, a Golgi body contains around 3 to 20 cisternae arranged one above another like stack plates. The Golgi apparatus is especially prominent in cells that actively synthesize and secrete proteins, such as gland cells. Structurally, the Golgi complex has two distinct faces. The CIS face, also called the receiving side, and the trans face, known as the shipping side. Transport vesicles coming from the endoplasmic reticulum fuse with the CIS face of the Golgi apparatus. Inside the Golgi body, proteins and lipids are modified, sorted, and packaged. After processing, new vesicles bud off from the trans face and carry the materials to their final destinations inside or outside the cell. The Golgi apparatus therefore acts like the packaging and distribution center of the cell. It helps in secretion, storage, transport, and formation of lysosomes and secretory vesicles. Golgi bodies also participate in the synthesis of certain carbohydrates and cell wall materials in plant cells. Ribosomes are known as the protein synthetic machinery of the cell because they are responsible for making proteins. They are tiny granular structures found either freely floating in the cytoplasm or attached to the surface of the rough endoplasmic reticulum. Ribosomes were first observed by George Palade in 1953, which is why they are sometimes called Palade particles. Structurally, each ribosome is made up of two subunits: a large subunit and a small subunit. Both subunits are composed of ribosomal RNA, also called rRNA, along with proteins. Ribosomes are non-membrane-bound organelles and are extremely small, measuring approximately 22 by 32 nanometers. Interestingly, ribosomes contain nearly 85% of the total RNA present in the cell. Now let's understand their functions. The two ribosomal subunits come together only during protein synthesis. Ribosomes read the genetic instructions carried by messenger RNA and join amino acids together to form proteins. Free ribosomes in the cytoplasm mainly synthesize proteins used inside the cell, while ribosomes attached to the rough synthesize proteins meant for secretion or membrane formation. The ribosomal RNA required for ribosome formation is synthesized inside the nucleus of the nucleus. Lysosomes are small vesicle-like organelles filled with powerful digestive enzymes. These enzymes help in intracellular digestion, meaning digestion that occurs inside the cell. Lysosomes break down unwanted materials, harmful bacteria, food particles, and even worn-out organelles. When a cell engulfs particles through a process called phagocytosis, a food vacuole is formed. This vacuole then fuses with the lysosome, and the digestive enzymes inside the lysosome break down the material into simpler substances. The useful substances are reused by the cell, while waste materials are removed. Lysosomes also perform autophagy, which means digesting old or damaged cell organelles and recycling their components. Because of this recycling function, lysosomes help maintain cellular cleanliness and health. Now let's look at the important functions of lysosomes. First, they help in garbage disposal by removing cellular waste. Second, they destroy bacteria and foreign particles entering the cell. Third, they digest worn-out organelles and recycle useful components. And finally, they play a major role in intracellular digestion and defense mechanisms of the cell. A vacuole is a membrane-bound sac present inside the cytoplasm of the cell. It mainly acts as a storage compartment and can contain liquids, dissolved substances, or even solid materials. Vacuoles are usually larger than vesicles and are especially prominent in plant cells. In plant cells, there is often one large central vacuole that occupies a major portion of the cell. This vacuole is filled with a fluid called cell sap, which contains water, sugars, salts, pigments, enzymes, and waste materials. The membrane surrounding the vacuole is known as the tonoplast. Now let's look at the functions of vacuoles. Vacuoles act as storage containers for water, food, enzymes, minerals, pigments, and waste products. They help maintain turgor pressure in plant cells, which keeps the plant firm and upright. Vacuoles also help in maintaining the osmotic balance and pH of the cell. In some organisms, vacuoles participate in digestion and removal of unwanted substances. Pigment stored in vacuoles can also give color to flowers and fruits. Centriole is a cylindrical cellular structure mainly found in animal cells and in many protists. It is located inside a specialized region of the cell called the centrosome, which acts as the microtubule organizing center of the cell. The primary role of centrioles is the production and organization of microtubules. Microtubules are tiny tubular protein structures that form part of the cytoskeleton of the cell. These microtubules perform several important functions such as maintaining cell shape, intracellular transport, movement of chromosomes during cell division, and formation of structures like cilia and flagella. Now let us understand the structure of a centriole. Each centriole is a short hollow cylinder made up of 9 triplets of microtubules arranged in a circular pattern. This arrangement is commonly called the 9+0 arrangement because there are 9 sets of microtubule triplets surrounding an empty central region. Usually, centrioles occur in pairs. The two centrioles are positioned at right angles to each other inside the centrosome. This arrangement becomes extremely important during cell division. Before a cell divides, centrioles duplicate and move to opposite poles of the cell. They help in the formation of spindle fibers, which pull chromosomes apart during mitosis and meiosis. Another interesting fact is that centrioles are structurally similar to the basal bodies of flagella and cilia. Basal bodies act as anchoring points for these locomotory structures. The cytoskeleton is a network of protein filaments spread throughout the cytoplasm. It helps maintain the shape of the cell, provides structural support, and assists in movement inside the cell. It is a dynamic structure attached to the plasma membrane and is made up of 3 main filament types: microtubules, intermediate filaments, and microfilaments. Among these, microtubules are the largest cytoskeletal components. They are long, hollow cylindrical structures made of proteins called alpha and beta tubulin. Microtubules perform several important functions. They maintain cell shape and structural support, help in the transport of organelles and vesicles, assist in exocytosis, form the mitotic spindle during cell division, and are involved in the movement of cilia and flagella. They are also major components of neurons, especially axons and dendrites. First, intermediate filaments. These are strong, rope-like protein fibers present inside the cell. They are thicker than microfilaments but thinner than microtubules. Intermediate filaments are made of proteins such as keratin, desmin, and neurofilaments. Their major function is to provide mechanical support and stability to the cell. They help cells remain strong and resist stress. They also assist in intercellular attachment, strengthening neurons, and maintaining the structure of skin and hair cells. Now let's move to microfilaments. Microfilaments are the thinnest fibers of the cytoskeleton. They are mainly composed of the protein actin. These filaments form a mesh-like network beneath the plasma membrane and help maintain the shape of the cell. Microfilaments play important roles in muscle contraction, cell division, cell movement, and formation of structures like microvilli. The cytoskeleton is a dynamic network of protein filaments spread throughout the cytoplasm. It helps maintain the cell's shape, provides structural support, and assists in movement inside the cell. There are three main types of cytoskeletal elements. First, microtubules. These are long, hollow tubes made of tubulin proteins. They help in chromosome movement during cell division, transport of organelles, and movement of cilia and flagella. Second, intermediate filaments. These provide mechanical strength and stability to cells. They are made of proteins like keratin and desmin and are especially important in skin and nerve cells. Third, microfilaments, the thinnest filaments, made of actin protein. They support cell shape and help in muscle contraction, cell movement, and cytokinesis. Peroxisomes, tiny but very important cell organelles. Peroxisomes are also called microbodies and are known as subcellular respiratory organelles. They are small membrane-bound structures found mainly in liver cells and kidney tubular cells. These organelles contain powerful enzymes such as oxidases and catalase. During metabolic reactions, peroxisomes produce hydrogen peroxide, or HO, as a byproduct. Since hydrogen peroxide is toxic, the enzyme catalase immediately breaks it down into water and oxygen, protecting the cell. Peroxisomes mainly help in the oxidation of amino acids and long-chain fatty acids. They also remove harmful reactive compounds from the cytoplasm and play a key role in detoxification. Spherosomes are small membrane-bound vesicles found only in plant cells. They are about 0.5 to 1 micron in diameter and mainly consist of lipids. Their primary function is lipid storage, helping plant cells store oils and fatty substances efficiently. Now, moving on to the nucleus, the control center of the cell. The nucleus contains the genetic information needed to regulate all cellular activities and chemical reactions. It is one of the most prominent features of eukaryotic cells. The term eukaryon actually means a true nucleus, highlighting the importance of a membrane-bound nucleus in eukaryotes. The nucleus stores the genetic information of the organism in the form of DNA. This genetic material controls all cellular activities. One of its major functions is controlling cell division, ensuring proper growth and reproduction of cells. The nucleus also directs protein synthesis by providing the template for RNA formation, which later helps in making proteins essential for the cell. Another important role of the nucleus is separating the DNA from the cytoplasm's complex metabolic activities. This protection helps maintain the integrity of genetic material. Finally, the nuclear membrane acts as a protective boundary, regulating the movement of substances between the nucleus and cytoplasm. The nucleus mainly consists of four parts: the nuclear envelope, nuclear plasm, nucleus, and chromatin. The nuclear envelope is a double membrane with tiny nuclear pores that regulate the movement of materials between the nucleus and cytoplasm. Inside the nucleus is the nuclear plasm, a fluid medium where nuclear components are suspended. The nucleus is a dense region rich in RNA and proteins. It plays a major role in ribosome formation. Chromatin consists of DNA and proteins, carrying the genetic information of the cell. Now looking at the nuclear envelope in detail, it has inner and outer membranes separated by the perinuclear space. The outer membrane is continuous with the endoplasmic reticulum and may contain ribosomes. The inner membrane is supported by the nuclear lamina, made of proteins called lamins, which help maintain nuclear shape and provide attachment sites for chromatin. The nuclear pores are small cylindrical channels present in the nuclear envelope. They create a direct connection between the cytoplasm and nucleoplasm. These pores regulate the movement of RNA, proteins, and other molecules in and out of the nucleus. Their number varies depending on the activity and type of the cell. Next is the nucleus, often called the ribosome factory of the cell. It is a large and dense structure inside the nucleus, but unlike many organelles, it does not have a membrane. The nucleus mainly contains fibrillar components made of DNA and RNA. Here, genes responsible for ribosomal RNA are actively transcribed and processed. The granular component contains RNA molecules and proteins that combine to form ribosomal subunits. These subunits are then exported to the cytoplasm, where they participate in protein synthesis. The size of the nucleus is directly related to the level of cellular activity. Cells with high rates of protein synthesis usually have very large nucleoli because they need many ribosomes. Now, during cell division, an important change occurs. Chromatin condenses into compact chromosomes, and the nucleus gradually shrinks and disappears. At this stage, RNA and nucleolar proteins disperse or get temporarily degraded. After mitosis is completed, chromatin uncoils again, nucleolar organizer regions become active, and RNA synthesis resumes. Eukaryotic chromosomes are mainly made up of two components, nucleic acids and proteins. The nucleic acid part is primarily DNA, which carries the genetic information of the cell. Small amounts of RNA are also present. The protein component mainly includes histones and non-histone proteins. Histones are positively charged proteins that tightly bind to negatively charged DNA, helping in DNA packaging and stability. Core histones include H2A, H2B, H3, and H4, while H1 acts as the linker histone. Under the electron microscope, chromatin appears like beads on a string. These bead-like units are called nucleosomes, which are formed when DNA wraps around histone proteins. A nucleosome is formed when about 146 base pairs of DNA wrap around a histone optima. This optima contains two copies each of the core histone proteins, H2A, H2B, H3, and H4. The DNA between two nucleosomes is called linker DNA, and histone H1 helps stabilize this structure. Under the microscope, chromatin looks like beads on a string, where each bead represents a nucleosome. As DNA packaging continues, these nucleosomes coil further into a 30 nanometer chromatin fiber, then loop and condense to finally form a fully packed mitotic chromosome during cell division. This remarkable packaging allows nearly 2 meters of DNA to fit efficiently inside the tiny nucleus of a cell while protecting genetic information and regulating gene expression. Chromatin is the complex of DNA and proteins present inside the nucleus. During interphase and early prophase, chromatin exists in two major forms, euchromatin and heterochromatin. Euchromatin is lightly stained and loosely packed. Because of its open structure, it is genetically active and allows transcription and gene expression. It also replicates earlier during the S phase and is generally GC-rich. On the other hand, heterochromatin is densely stained and tightly packed. It remains highly condensed throughout the cell cycle and is usually genetically inactive. These regions replicate later during the S phase and are often AT-rich. Think of the cell as a tiny living factory, where each organelle has a specialized job. The cell wall, membrane, and cytoskeleton provide support and shape to the cell. The cell membrane also controls what enters and leaves the cell. The endoplasmic reticulum works like an internal transport system, moving materials throughout the cell. Mitochondria are called the powerhouse because they produce energy. The nucleus acts as the control center, storing genetic information and directing cell activities. Ribosomes, and chloroplasts help in producing important cellular products like proteins and food. The Golgi apparatus functions as a packaging and shipping center, while vesicles transport materials out of the cell. Vacuoles store water, food, and waste materials. Lysosomes and peroxisomes act as recycling centers by breaking down waste and harmful substances. Chloroplasts convert light energy into chemical energy through photosynthesis. Finally, structures like nuclear DNA and centrioles help in forming new cells during cell division. Together, all these organelles work in perfect coordination to keep the cell alive and functioning efficiently. Thank you so much for watching this video. I hope this presentation helped you understand the fascinating world of cell organelles and their functions in a simple and interesting way. If you enjoyed the video and learned something new, don't forget to like, share, and subscribe to the channel for more biology and science content. Leave your questions or suggestions in the comments section below. And make sure to hit the bell icon so you never miss future updates. Your support truly means a lot. See you in the next video.